This is the first lesson of Chapter 5, where we're going to be talking about plasma membrane structure and function. Specifically, in this lesson, we'll focus on the structure. Before we look at the components of the plasma membrane, some of which are shown in the illustration here for an animal cell, let's first just talk in general about what is the plasma membrane. You might recall from the previous chapter, Chapter 4, an introduction to the cell and the cell parts, that we learned that the plasma membrane is the boundary that separates the living cell from its non-living surroundings. Plasma membranes are also known by other names, so if you perhaps see or hear me use this term, I'm using them interchangeably with one another. So the cell membrane would be another term for the plasma membrane. Sometimes folks might also refer to it as the lipid bilayer, which is more what it's made of partly. Predominantly, it is a two-layer structure of lipids, or more specifically, even phospholipids. So just be familiar with the different terms that might be used, but for the textbook that we use in this class, the plasma membrane is the vocabulary term. So because this is the boundary around the cell, separating it from its non-living surroundings, this membrane plays a significant role in the passage of materials in and out of the cell, or also preventing certain materials from passing in or out of the cell. So this property of allowing certain things to pass and other things not is called selective permeability. So we could say that the plasma membrane is selectively permeable, which means certain substances can cross more easily than others. In general, the ones that can cross more easily are small, hydrophobic, nonpolar molecules, such as hydrocarbons or gases, would be another example. Gases like carbon dioxide and oxygen. Let me add a note here that these are also generally small in size. So small hydrophobic nonpolar molecules are preferred to go directly through the plasma membrane versus large polar molecules such as sugars cannot cross the membrane easily. But sugars are food, and some organisms and cells do need to be able to obtain that food and possibly release the waste that is the byproduct of the digestion of that food. So it's not that they can't get in, it's just that they need to have the aid of a structure to be able to pass in and out of the cell for those large polar molecules. And we'll look more at the process of how these materials pass through the cell membrane in our second lesson of Chapter 5. So for now, we're going to focus now on the components of the plasma membrane. One way to describe the plasma membrane is to say that it is a fluid mosaic. Fluid mosaic model of a plasma membrane refers to the fact that it it has fluidity. It's able to move. It's pliable. It's not like a brick wall that doesn't allow anything to pass. As we've just actually discussed, we know that things can pass through the lipid bilayer directly, or perhaps through the aid of a protein channel, like these purple, they're meant to represent proteins that are embedded in the membrane. So we'll get more into the fluidity in a few minutes as well, about the fact that the plasma membrane is it moves, it's pliable, and there's some interesting things that happen, in fact, with the movement of the phospholipids. But on this slide, I want to emphasize more the mosaic aspect of the plasma membrane. So when you hear the word mosaic, does something come to mind? I think back to an art project that I did when I was in elementary school, where we were given a bunch of different pieces of crushed tile or pottery, and a square piece of cardboard and some like concrete mud, and we were making a new piece of art with those broken pieces of pots um, and tiles. So what a mosaic is like a conglomeration or a collection of different components to make something. So that something here is the plasma membrane. And as you look at this illustration, it's kind of overwhelming how much is there. So many different structures of the plasma membrane. Now this is an animal cell, so there are more things going on with the extracellular side of the animal cell, but I want to fixate mostly on what's happening at the membrane. So I'm drawing your attention here to the predominant components of the plasma membrane, which are the lipids or phospholipids. Remember, they have the two main parts, the phospholipid head, those gray ovals, and then the fatty acid tails, two of them attached to the phosphate group. So it's mostly that, phospholipids, and then secondarily you have these proteins. Some of them are embedded in the membrane and some of them are attached to either side of the membrane. The integral proteins are embedded in the membrane and the peripheral proteins are on either side of the membrane. They don't span the full diameter or distance of the membrane. Now these have different roles, and we'll talk more about that when we get to the second lesson of chapter 5. What is the function of these membranes, specifically in transport of materials in and out of the cell? So let's start more with digging in deeper to the primary components of the plasma membrane, which are the phospholipids. Oops, went a little too far there. Okay, so zooming in here to a phospholipid bilayer, you can see that the fatty acid tails are all facing inward. Remember, they're hydrophobic, so they're repelled from water and they assemble in this manner just because of their nature being nonpolar, hydrophobic. They get away from the water, which means the hydrophilic heads, which are the phosphate groups, can interact with water. So this has a property that's termed amphipathic, when you have a molecule that has both a hydrophilic region and a hydrophobic region. Okay, but remember that there's also proteins that can be embedded in the membrane. And in order for them to be there, to stay in this mosaic, if you will, of the plasma membrane, they also have to have that same property of being amphipathic, having a hydrophilic and a hydrophobic region. The hydrophobic regions are going to line up with the hydrophobic tails of the phospholipid bilayer, and the hydrophilic regions are going to be on the outer and inner surfaces. So the interior of the cell and the exterior of the cell is where the hydrophilic regions of the proteins would be. So bear in mind that, and this kind of gets more at the fluid nature of things, so that these are just being held together by weak hydrophobic interactions. Just the fact that all the hydrophobic things are trying to get away from the water, the aqueous surroundings. This is not a strong bond in any way. And so easily this can disassemble, and there's also fluidity within the plasma membrane because of the nature of these weak hydrophobic interactions. In addition to the fact that they're weakly held together and so things can move through, the components of the plasma membrane can also literally move within the membrane. They can swap places with their neighbors right next to them on the same layer of the bilayer. And they can also swap places with their neighbor across on the other bilayer. Now that doesn't happen as often, but point being that nothing is static here. There's fluidity, and this in turn will influence the ability of materials to pass through the membrane. The environment can also influence the fluidity of the membrane. One environmental factor that has a big impact on the fluidity of the membrane is temperature. As you might recall from when we talked about properties of water, or maybe you know this from chemistry and physics as well, molecules at higher temperatures have more kinetic energy. The random particle motion is going to be faster at higher, warmer temperatures. And versus at lower temperatures, the molecules are going to move more slowly, possibly even stop moving at all altogether if it's frozen. So this can in turn affect the fluidity of the membrane and its ability to allow materials to pass through it. A membrane must be fluid to work properly. So if it freezes and nothing can pass, then that cell or organism that cell is in could potentially die. So, but you might be thinking, well, there's animals that and plants and organisms that live in really cold places where temperatures get freezing. How can they survive? Well, one adaptation they have is in their plasma membranes. Recall that we talked about the different kinds of fats, saturated versus unsaturated, and that saturated fats are saturated fully with hydrogens on those hydrocarbon tails. So that would be the illustration here shown on the right. This is a saturated fat. On the left would be unsaturated. Now, these are, although we were talking about it previously in chapter 3 with respect to like food and health, if this is part of the plasma membrane, these could be the phospholipid tails. They could be saturated or unsaturated. And what difference is this going to have on that organism's ability to survive and maintain that membrane fluidity? Well, if the hydrocarbon tails of the fatty acids are of the phospholipids rather are unsaturated, you're going to get more kinks, and those give essentially more wiggle room between the phospholipids of the bilayer. Materials are going to still be able to pass through with all of those extra gaps between the neighbors, versus in a saturated, if they're all saturated as shown on the right, in a warm environment, it'll be all right because they still have a lot of kinetic energy to move through. So in a warm environment, there's going to be potentially an adaptation of having more saturated hydrocarbon tails of the phospholipid bilayer versus in colder climates where the temperatures aren't really causing the movement of those phospholipids. So that's why they need that added adaptation of the unsaturated fatty acid tails. There's another adaptation that has been seen in the membranes of animals that live in different climates, and that's the presence of the steroid cholesterol. And that's shown here in the membrane in both bilayers as the 4 carbon rings fuse together. So it serves really more like a temperature buffer, which means that in different scenarios, it helps to compensate for the environmental changes in temperature compared to inside the organism. So in warm temperatures, the presence of this cholesterol in the bilayer can restrain the movement of the phospholipids, kind of act like a barrier. The phospholipids next to those cholesterol molecules might be wiggling around in warm temperatures, but they're not going out of control because there's a barrier there to kind of still keep them in place. In cold temperatures, the cholesterol can also be a benefit because it helps maintain some fluidity. Notice there's a bit more of like gaps around the cholesterol compared to if it was all just fully saturated hydrocarbons. So you can't really say that like cholesterol is better in certain situations than others. It really depends on the organism. And we'll see an example of that in class when we look at an abstract of a paper about Alaskan wood frogs. If you're curious about that, the abstract of the paper is posted on the D2L course. Okay, so just to circle back and kind of bring us to where we're going to start our next lesson, recall that the membrane is a fluid mosaic. We looked at the main components, the phospholipids and the proteins. And to get us ready for the next lesson, you'll need to remind yourself, and so we'll do that right now, that there's the 2 different kinds of proteins. The integral proteins, they span through the membrane. They have the amphipathic property, having a hydrophobic interior, hydrophilic exterior, and then the peripheral proteins are loosely bound to the surface on either side of the membrane. And that'll leave us for next time. Continue.